Hello everyone. It's a great pleasure for me to introduce our speaker today, Dr. Gilles Vandewalle. He has PhD in neuroscience and a co-director of GIGA CRC in vivo imaging at University of Liège. His main focus is on the brain mechanisms involved in regulation of sleep, and also the link between sleep cognition and brain health using variety of techniques such as MRI, TMS EEG, and also PET scan. And today he will talk about the investigation of the link between the locus coeruleus activity during wakefulness and sleep electrophysiology using ultra high field MRI. I suppose it's 7 Tesla MRI that they have here. And yeah, at the end of the talk, we can have question and answers. We have, I think, 15 minutes to discuss. Thank you, Gilles, for accepting my invitation and joining us. The stage is yours. Thanks.
Okay, hello everyone. First, thank you, Masoud, for the introduction and for the invitation to give a talk as part of the ENIGMA-Sleep initiative. We can make this like very informal. So I see a bit of you and I can hear. So if you have burning questions, please feel free to ask questions while I speak. I don't know how long this presentation will be. It may be a bit shorter than usual, but it really gathers our most recent research interest and research findings. So as Masoud said, we are trying to look at the locus coeruleus and using 7 Tesla MRI. We do that during wakefulness and we try to link that with sleep, and I'll tell you what we did on that topic. So something, a really very, very broad message. First, I'm sure you're all aware of all this, but this is kind of a very broad introduction as to why one would be interested in studying sleep and trying to understand its variability. It's because sleep is stable in an individual and yet quite variable across individuals, and within an individual it changes within the lifetime and over the lifetime. And so I mean, it's good to understand why it is so and what are the implications of this, notably because, well, as we get older, we complain more about our sleep and we may develop insomnia. And sleep may also contribute to many diseases, including Alzheimer's disease, for instance, here. So you probably all know a lot about the locus coeruleus, but still, again, as an introduction and as to why we are interested in that structure. Well, first, it's the main source of norepinephrine of the organism, maybe the only one. It's involved in many things, in anxiety, in aspects of cognition, but also it's involved in sleep wake regulation. And because we are interested into the link between sleep and health and changes over the lifetime, we look at this also from this perspective and see that Alzheimer's disease see the first tau deposit in the LC or around the LC, in the LC and other structures of the brainstem. And the locus coeruleus may also contribute to insomnia. So it's an interesting structure to consider. One issue we may have when we work on the sleep of human beings is that the small scheme that I included up here, which shows the locus coeruleus at the back of the brainstem and some of its main projections at the subcortical level to form the what we know about the circuitry of sleep-wake regulation. All of this, or most of this, has been shown in animals. And by animals, I mean nocturnal rodents, which are quite different from human beings, notably in terms of cognitive functions they can have, development of the cortex, presence of melatonin, or also being nocturnal or diurnal. And so in fact, in humans, we don't know as much as one would think about the role of all these nuclei and how they shape the variability of sleep. And so all these nuclei are interested here, but today we will focus and try to address some of the aspects related to the locus coeruleus. A few things to know about the locus coeruleus, and that will be useful for the end of the talk, is that it's a small structure that can present two types of activity. Activity may be phasic and tonic, and this is thought to be along the line of an inverted U shape, where if you have in an ideal case, up on top here, you'd have an alternance between tonic activity of the LC, which is regular, let's say, low intensity firing of the locus coeruleus, and some phasic activity, for instance, in response to a stimulation outside, which would warn us about the presence of a stimulation in the environment. And the balance between tonic and phasic activity is important for the functions that are under the control of the locus coeruleus. Of course, you can go to the other parts of the inverted U shape and go to the left or to the right. You will mostly have a tonic activity, which could be either really low or really high, and wouldn't allow or wouldn't present at least some phasic activity. And you could be in hyperarousal on the one end, or in really low arousal on the left side. And so it's interesting structure in terms of its activity for this. Then the locus coeruleus is thought to evolve over the lifetime and have an integrity which changes. And people believe that if you make a particular sequence, MRI sequence, that allows you to detect something that's related to neuromelanin, which is a pigment that's present in the locus coeruleus, and which is thought to contain a lot of iron, and because of that could be detected with certain sequence in MRI. If you use such a neuromelanin sensitive sequence and contrast the signal you get from the LC to its surrounding, you have the so-called LC contrast, and people think it's related to the integrity of the structure, but it also follows some sort of inverted U shape, and the contrast may increase up until about 60 years old, and then would decrease due to degeneration after about 60 years old. And the implication of this increase in contrast and then decrease are still under investigation now. The LC is also very challenging to image in vivo. It's small, as I mentioned, and I didn't underline this. It's a kind of a small spaghetti shape of about 15 to 20 millimeter structure at the back of the brainstem. So it's small. It's also near the ventricle and quite deep in the brain, and so it's not easy to image. With the development of 7 Tesla MRI, it becomes more accessible, let's say. I wouldn't say it's easy, but it's more accessible. And so there are more studies interested in that structure now that 7 Tesla MRI is more available. A challenge also related to being interested in the locus coeruleus is that during sleep, its activity, if we look at the activity of the LC here, is getting lower during slow wave sleep, and it is considered to be silent during REM sleep. And so, of course, it is difficult to image, at least functionally, a structure that is silent. And so it is a challenge to record sleep in an MRI, and it will be a challenge to record the LC in an MRI, particularly if you're asleep. Reason why we decided here to look at the activity of the LC during wakefulness first, before maybe investigating whether we are able to pick up some of the lower amplitude signal that is thought to be triggered by the LC during sleep. And so our current question with the data we have accumulated so far is to assess whether the function of the LC and its variability during wakefulness would reflect variability in LC-controlled processes. And if so, then this would include sleep. And so we could potentially look at LC activity during wakefulness as a more accessible way to look at its activity when it's supposed to be active, and put that in relation with sleep. And so that's the main framework in which we are now. And we are trying, therefore, to investigate the link between the activity of the LC in vivo and some canonical sleep metrics in healthy younger and older individuals using both the EEG and 7 Tesla MRI. This is the current sample size. So we now have 52 individuals acquired. And we have a bit more younger individuals than older individuals. And so by younger, we mean from 18 to about 30 years old. And we have 33 of these individuals. And older, we are at 19 individuals at the moment. But by older, we mean late middle-aged individual, because when you get closer to 50, as I am, you don't see 50 as being old anymore. So it's still quite young. And so from 50 to 70, you could see this as a late middle age rather than being really into the last part of life, let's say. You have some of the exclusion criteria here, just for you to know that these people were devoted of sleep issues and had no cognitive impairments. This is the overview of the current protocol. We'll not look at all this during the presentation today, but participants have a screening night and habituation night, during which we look for sleep apnea and periodic movements in limbs. This is followed by structural MRI, which also is used as habituation to the scanner environment. Then about a week later, people come back to the center, and they have slept regularly for 1 week before they come to the lab. They do a baseline night of sleep, and then do a functional MRI in the morning. We also include some cognitive tests, and I will not come back to this now. So today we will be mainly focused on functional MRI and the baseline night of sleep. And as I will show, we need some of the data we acquired during the structural scans. For the functional MRI, we had 2 tasks. And I'll first describe the first one and the results we obtained with that task. This is the work by a former PhD of the lab who recently graduated, Katia Koshmanova. And the reason why we are using this auditory task, but also the next one, is because it is believed to recruit locus coeruleus activity and to trigger activity of the locus coeruleus. So the task we use is a mismatch negativity task, or the so-called oddball task, where you hear a stream of standard tones, always the same, and every now and then you have a deviant tone, which triggers which is detected by the central nervous system, and you have to press a button when you hear the sound. And so the task mimics novelty and salience. There is some previous data saying that it recruits the locus coeruleus. Sorry, I have something in the way on my screen, but yeah, I'll try to do like that. Okay, and so we also have, and that's the only time I will discuss structural MRI, with the neuromelanin sensitive sequence that would allow us to make individual masks of the LC. So there are several ways of detecting the LC. What we did is that we individually delineated the LC on each images, and I keep on forgetting to add an image where you actually see the raw signal and then see the mask on top of it, but you'll have to take my word for it. Just behind this yellow line, there is a slightly more hyper-intense line, which corresponds to the LC. This was done by 2 raters, and we only looked at the intersection of 2 raters, and it's still considered as the gold standard way to delineate the LC on MRI, although there are some automatic techniques being developed. And so this allows us to, for each individual, extract the activity of the LC, or at the group level to create a probabilistic template of the LC to assess whether the activations we may see are within the LC or not. This is the results of the oddball task. And for those who have worked with the oddball task before, you know that it recruits many areas in the brain, and that's what you see here. There are many brain structures involved in the detection of salience. Of course, what we are interested in is to see whether some of these voxels are part of our locus coeruleus probabilistic template, and it's the case. And so you see here the small activation we get in the LC. It's also a local maximum, so it's not just a leak from the surrounding structure within the LC, but there is a local source of bold variation, at least on the left side of the brain for the locus coeruleus. And if we do a correction over the probabilistic template, we can argue that we get something significant. In any case, the purpose of this is not to get, per se, a significant activation of the LC. It's to have the impression that something is going on in the LC, so that we feel that we have the right to go at the individual level to extract activity of the LC and then to relate that to some sleep metrics. What are the sleep metrics we extracted from the baseline night of sleep? The first, we looked at subjective sleep quality with the Pittsburgh Sleep Quality Index, just to get an overview of how sleep is perceived. This is the only subjective measure we used in the analysis so far. And then we took some metrics which we think are a limited set of metrics that reflects many aspects of sleep, because of course the more metrics you put, the more issues with multiple comparisons you get. And so we looked at sleep latency as how well you are able to initiate sleep, then sleep efficiency, then overall structure of sleep, which is the percentage of REM sleep, which is also the percentage of slow wave sleep in a way. And then we quantified slow wave activity, but in a cumulative way over the night. And in such a case, it's called slow wave energy. So it's the cumulative delta power of the night from 0.5 to 4 Hertz during non-REM sleep. And to have a kind of a mirror, another metric that would be quite equivalent for REM sleep, we looked at theta activity power during REM sleep, theta being probably the most typical rhythm of REM sleep. And we also looked at it in a cumulative way. So we took the cumulative power of REM sleep overnight. So this is for the sleep metrics we considered. We extracted the activity of the LC during wakefulness during the oddball task, and we then tried to relate the activity to what we were measuring. So first we looked at sleep quality, subjective sleep quality, the lower value meaning the best sleep quality. And when we take the activity of the LC as a regressor for sleep quality, we see that sleep quality, subjective sleep quality, is related to LC activity, but differently with age, and we get a significant interaction. And when we do post-hoc contrasts, we find that it's actually only significant in older individuals. And when we look at the data, we see that the higher the activity during wakefulness, the poorer is subjective sleep quality. So if you have a more active locus coeruleus activity during wakefulness, then you have a worse sleep during the protocol. Of course, we were asked by reviewers to also look at LC contrasts. And when we do that, we find a relationship that we haven't pursued too much so far, is that the contrast of the LC is also related to subjective sleep quality. And the higher the contrast, the better the sleep quality. But this is true irrespective of age, so for both older and younger individuals. And I realized that, sorry, I should have mentioned, so in blue here is the older individuals, and in orange are the younger individuals. And so I could have changed that. So here the relationship depends on age. Here it doesn't depend on age. Importantly, if we control for LC contrast in what I showed you here on the left side of the slide, the interaction and the effect in older individuals remains significant. That was for subjective sleep quality. If we now move to objective measure of sleep, the only measure for which we find a significant relationship is when we look at REM sleep cumulated theta power, so between 4 and 8 hertz. And we find that the higher the activity of the LC, the lower the intensity of theta power during REM sleep. And this is true in older individuals, and there is no significant relationship in younger individuals. Again, this result was a bit challenged by the reviewer we had. And so if we remove what could be called outliers in the young individuals, and this is based on Cook's distance, which could be considered as too high for these two individuals, then the p-value gets better and not worse. So it's reason why we think that this relationship in older individual is quite robust, even though we would want it to be replicated, and we would want to see a p-value that's lower than that. Importantly, if we look at the other metrics I mentioned before, sleep onset latency, sleep efficiency, REM sleep percentage, or slow wave energy, as well as the other frequency bands during REM sleep, we don't find any significant association. So it seems that the association between the activity of the locus coeruleus during wakefulness, during the oddball, with sleep, it's only present for theta power of REM sleep, and it seems to be specific to that. So at the first summary, this tells us that we can associate activity of the LC during wakefulness with some sleep metrics, either subjective sleep quality or the intensity, or a marker of the intensity of REM sleep. And that would be particularly present in late middle-aged individuals. We thought that maybe it is the reflection that if the activity of the LC is higher during wakefulness, or is more reactive, more excitable, the LC may also be less silent during sleep, and that it would be because of that we would see an effect on sleep quality. And so we did a few more analysis on this, and we were able to show that subjective sleep quality is in fact indeed related to theta power in REM sleep. This is not a big surprise. There are some other publications showing this, that subjective sleep quality is related to REM sleep. So we were relatively happy to see that, because it could support that the activity of the LC disturbs REM sleep to affect sleep quality. But continuing on this, we tried a mediation analysis. Of course, we only used the 19 older individuals for this, but we didn't find any significant results. So we couldn't support what we found, maybe because of sensitivity, or maybe because it's not what's going on. But if we look at the direct path, we find a significant direct path between the locus activity and subjective sleep quality. But if we go through the indirect pathway, it's not significant in the analysis we run with such a limited sample size. So we will need more subjects maybe to try again this analysis. Okay, so good, and this was recently published, so we are all happy about this. And that's oh, sorry. Yeah, I will come back to that in a second. And that's when we move to the other task, and that's when things become a little bit more complicated, at least to us. and maybe you'll have good suggestions as to why we see what we see with the other task. Coming back to this conclusion, maybe what we are looking at healthy individuals in this, and they are not insomniacs and they don't have any sleep disorders. And maybe if the activity of the LC gets even worse or higher during wakefulness, sleep quality will get also even worse. And maybe we are looking at things as part of a healthy continuum towards insomnia, but that's just a guess or a hypothesis. And I must acknowledge that there are some limitations to what we do, except for the sample size, which I've mentioned already, is that the older individuals did the sleep and the MRI acquisition, the EEG and the MRI acquisition up to 1 year apart. As to the young individuals, it was always done the baseline night followed by the MRI acquisition. We are unsure how that could explain the results we find, but it could bias in part what we did and contribute or mask also part of the things we could have found with this data set.